So what do we have here? We have here a cross section of a dicot stem. So as you can see here, these acorn shaped structures are vascular bundles. In the vascular bundle, interiorly you have the primary xylem, exteriorly you have the primary phloem. In between this primary xylem and primary phloem, you have the vascular cambium. When it's time for secondary growth, what happens? The parenchymal cells that are found over here, they undergo modification such that they get meristematic characteristics, eventually forming a ring. So I'm not going to draw the entire thing. Right. You get a structure like this eventually. Okay. So this entire ring structure that you see here is known as the vascular cambium. So there are two meristems that are involved in secondary growth. One is the vascular cambium, the other one is the cork cambium. In the vascular cambium, you have meristematic cells. We also call them as initials. So we can also call this as a ring of initials. In this ring of initials or these meristematic cells, some of them are oriented perpendicularly to the long axis. So if you take this as the stem, it is extending this way, no? So this is the axis. So if the cells are oriented in this manner, that is perpendicular. If the cells are oriented in this manner, then that is parallel. The cells that are oriented parallelly involved in the formation of the secondary xylem and the secondary phloem. So if it is forming secondary xylem, what are the cells that it forms? See, vessel elements, tracheids, parenchyma and fibers. If it is phloem, then sieve tube elements, companion cells, parenchyma and fibers. So which direction does it form the primary xylem? Which direction does it form the secondary xylem? Which direction does it form the secondary phloem? Secondary xylem is produced inwards, secondary phloem is produced outwards. Right. So this forms secondary xylem inwards, secondary phloem outwards. So far we only spoke about those initials or meristematic cells that are oriented parallelly to the long axis. There are also initials that are oriented perpendicular to the long axis. These, what do? They form something known as vascular rays made up of parenchymal cells. These parenchymal cells function in connecting this secondary xylem and the secondary phloem, or connecting the xylem and the phloem. In addition to that, it is also involved in storage and wound repair. Is that clear? Right. So due to this, circumference can increase. Then that is the entire function of the vascular cambium. Is there anything else? No, that is it. Then we will move on to the cork cambium. This cork cambium is a meristematic cell layer. It can be one cell, 2, 3, 4, 5, right? So like we saw previously, how these parenchymal cells can undergo modifications to form vascular cambium, the parenchymal cells here undergo modification to form the cork cambium. This area of the ground tissue is known as the cortex. So parenchymal cells in the cortex can undergo modification to form this cork cambium. So what does the cork cambium do? Cork cambium produces cells outwards and inwards. Outwards and inwards. Outwards, what does it produce? Cork cells. Inwards, it produces parenchyma. You don't need to know that. Then, due to the cell production, what happens to the circumference? Increases. Just like a fat man wearing his 6 months old clothes, then what will happen to the epidermis? It will split and dry and fall off. Okay. As the circumference is increasing, as the action of the vascular cambium and the cork cambium is increasing, what happens? This secondary xylem continues to accumulate. Secondary phloem continues to accumulate. Cork cambium produces cork cells, continue to accumulate. Then, secondary xylem that accumulates is known as wood. The cork cambium and the cells it produces collectively are known as the periderm. So now, due to this process happening continuously, even the cork cambium is slowly pushed out. Yes? So as the cork cambium is slowly pushed out, eventually even the cork cambium can become cork cells and it also will split and fall out. then at that point there won't be any cork cambium. Then secondary growth will stop. But does secondary growth stop? No, it is continuous. Then what happens? Just like how previously from parenchymal cells cork cambium was able to form, again from parenchymal cells cork cambium will form, and that process will continue. Okay. All the cells that are produced from this cork cambium are known as cork cells. Sorry, the cells that are produced exteriorly from the cork cambium is known as cork cells. They undergo a special type of modification where they deposit suberin in themselves, making them dead. However, they are at functional maturity, they are dead. This protects the entire tissues that are present inside the stem from external factors, from water loss, from infection. So many different factors these cork cells can protect from. However, keep in mind these cork cells are impermeable to water and gases. But there needs to be some sort of gas exchange between the internal and the outside. That's where these special structures known as lenticles come into play. Even though these cork cells are tightly arranged in many areas, there are certain places where cork cells are loosely arranged. That will form a horizontal slit, which I will show later, known as lenticles. So in epidermis, we had something known as stomata. Over here we have lenticles. Right. So this process continues throughout. Eventually, all the tissue, all the substances that are found exterior to the vascular cambium is known as the bark. Secondary xylem is known as the wood. Cork cells and the cells cork cambium and the cells it produces is known as the periderm. So the main component of bark would be secondary phloem and the periderm. So that is secondary growth. Is that clear? Eventually, finally, we get a structure like this. So that is a summary. Okay.